Okay, I know I gave you a promise, and I am falling on my word because I told you that in this video we were going to go on to our next, which is TLC. But before I do that, I want to clear some things up with the electrophoresis because it might have went under the radar or I just simply forgot to mention it. So first, I do want you to kind of keep in mind some of the terminology that they might use with electrophoresis chambers. So we said that we had this kind of galvanic cell setup, and during the electrochemistry lecture, we kind of gave names to these compartments. We gave names to the negative charged compartment and the positive charged compartment. So I want you to be very careful with the terminology because this is something that a lot of people get backward. So this negative end, well, the negative end is called the cathode. So some of these questions might want to know which one would move closer to the cathode or which one would be moving closer to the anode. So that is what we label the cathode and anode as. You got to keep in mind it is negative. The negative end attracts the cations, which is why it's called the cathode, right? Cation, cathode. Well, the same thing holds true to the positive end. The positive end attracts the anions in solution, so we call it the anode for that reason. So that's going to be one of the most confusing things that you will have to happen to you in these problems, is it will say which ones will migrate to the cathode, which ones would migrate further to the anode, and a lot of people get them backward because they don't think about it that way. Cathode attracts the cations. So in order to attract the cations, you have to have a negative pole in place. So the negative end is called the cathode. The positive end is called the anode. So that's number one. Number two, something else that I probably didn't follow through on in the previous video, was that this is buffer pH dependent. All right, so what that basically is going to tell you is that the buffer has control of the entire thing. And a lot of times we work with buffers that are somewhat neutral. So buffers of pHs of around 5, 6, 7, 8, somewhere in there is where we're working with the buffers. But they could get a little tricky on you and they could kind of change the buffer up a little bit. And when they change the buffer up, then this separation process can change slightly as well. So what you have to look at is you're comparing the pI of the amino acids to the pH of the buffer that we are using in solution. And what you will see is that the more positive the amino acid will be, and the farther it will migrate toward the cathode in a given amount of time, if the pI and the pH are far apart. So if I take the difference, so let's come down here and let's say the difference between the pI and the pH is great. So a lot. The farther the amino acid's pI is from the pH of the buffer, that means it's more positive and it will go toward the cathode end. So if I look at these pI values that we did in our example, we had a 10.6. Well, let's say that this pH that they used was a 5. Okay, so 10.6 is the furthest away from 5, so that means it's way over here toward the cathode. And then 6.02 kind of comes in next, 2.98 kind of comes in next, but that's how you would order that separation process. So, take a look at the pHs. Understand that the pH of the buffer is very important. The further away that those PIs are, the more positive that they are, and if they're positive, then they go toward the cathode. If it's lower than the pH, then the more lower that they are, the further it goes to the anode. Right? So the same thing on both ends. So pH of 5 is kind of the dividing line. So that means that if this was kind of the middle of the sheet of paper, and let's say this is the neutral area. So this is where neutrality is at. Let's just pretend that's what happened here. Well, 10.6 is higher than 5, which means it's more positive, so it's over here to the left. 6.02 is higher than 5, that means it's more positive, so it's over here to the left. 2.98 is lower than 5, which means it's kind of more negative, so it's over here to the right. And that is kind of how you would order those amino acid separations. Typically, it goes in order of the PI values. That is how they separate. But just to make sure that everybody is really fully understanding what's happening on that sheet of paper, it is pH dependent. You look at the PI values. The further away that they are, the more that they will travel, because the more positive or the more negative that they are. So let's say that we had an amino acid out here that had a PI of a 1.3. Where would that fall? 1.3 is lower than 5, right? So if I put them in order, 10.76, 6.02, 2.98, 1.5 would probably be over here, and that would be right. So you can kind of see how just looking at the PI values, they will fall in order. But just to kind of make sure that we understand it is somewhat buffer dependent, that is what we're doing. So pH of 5 is kind of the cutoff. 1.3 is much lower than 5, so that means it will be negative, and that means it will go toward the anode, or the positive charge on the electrophoresis chamber. So just a few points that I wanted to make sure that we all kind of understood before we went on. Another thing that maybe we need to mention at least is these spots. Once the amino acids will spot out the way that they do, these spots, well, you won't be able to see them. Right? And the reason that you won't be able to see them is because amino acids have no color. So amino acids are colorless. So we've got to have maybe something in place where we will be able to see the amino acids and where they fall. And this requires indicator. So indicator is going to be very important, and the most common indicator here is ninhydrin. So if you've used an amino acid solution in our lab, that is probably how we isolated and identified the amino acids. We use ninhydrin indicator. And what happens is that it turns this stuff purple. So I would see a spot here, and I would see a spot there, and I would see a spot here, and I would see a spot there. So I would spray this ninhydrin on the sheet of paper. Wherever those amino acids would show up, we would see a purple area that would be there, and we would know that would be the presence of an amino acid. This is how they do your fingerprints on crime scenes and that kind of thing. If they're dusting or trying to lift for a fingerprint, your fingerprints are basically made up of some amino acids, and they can take a look at a fingerprint, spray it with ninhydrin if they wanted, and then allow that ninhydrin to develop, and the area of your fingerprint houses the amino acids, and that's how they can get these images of fingerprints on crime scenes and CSI if you're a fan of those shows. But this is basically the same type of thing. You could take your fingerprint, put it on the sheet of paper that you're putting the amino acid on, and if you sprayed it, you would see your fingerprint show up on that sheet of paper. So the same exact thing happens, which is why typically when you work with protein solutions, you wear gloves. You don't want to be contaminating the paper or the electrophoresis chamber or whatever you're using for the analysis. because you can contaminate these samples with your own amino acids, and that's something that we don't want to happen. So gloves are kind of important when you work with amino acids for that reason. And then hydrin is this purple colored product that makes it show up. So with that said, we now can talk about TLC, and that is what we'll do in the next video. I promise I won't let you down this time.